Okay. So when we left off, we'd been walking our way through the renal system. We talked about all the different uh, directions fluids can flow. We talked about reabsorption, secretion, excretion. We'd moved on to this idea of water balance, which is about where we left off. Some of the basics about input of water to our system and elimination output of water from the system, talking about how the kidney critically sets up this osmotic gradient in the medulla so that we have enough of an osmotic pull on water to do regulated reabsorption, which is largely what we'll be talking about today. Okay. So just to recap, as we were thinking about that osmolarity, so we go deep into the kidney. Well, let's remember uh, what our osmolarity was like in those deepest parts of the kidney and which part of our nephron, one of the pieces of the loop of Henle, is primarily responsible for creating this gradient. Let's take a look. Okay, cool. So we got for sure that we have high osmolarity in the deepest parts of the medulla, right? So this is a high pull on water. So we got that part good. We got a little more looking at that structure of the loop of Henle to do if you got this one wrong. So for our descending limb of the loop of Henle, this is where we initially had some water movement possible. It's the ascending limb where we are pumping solutes. Okay, so this is creating this gradient from the ascending limb. Okay, so last time we talked about that establishment, and I know we didn't record because of our technical issues, so I'll find a good short recording of that to put up. But we're going to move forward to thinking about how we use this medullary osmotic gradient, right? So these next two slides are just a couple of extra details about that gradient. Okay, so one of which is that it's not just that active pumping of solutes that helps us maintain this strong pull on water, this strong osmolarity of the medulla, right? We also have this waste product called urea. So this is something that's generated by the liver. It's for nitrogen elimination, really. So like as you're breaking down proteins and stuff. This is a substance that is really water soluble, but it does require transporters to move across the cell layers that we're looking at here. So you can see the location of these urea transporters in the image. And one of the things to note here is that some of that high osmolarity that we see in the medulla in these deeper parts of the kidney are because we also have movement of urea into those areas. So this is like an add-on here to what we discussed last time. Okay. The final little detail to add on to that medullary osmotic gradient is the role of blood flow. So the vasa recta are these straight up and down capillaries that we can see along the loop of Henle. Okay. And the way that they're arranged looks so funny or looks so structured, kind of mirroring that loop of Henle structure, because these capillaries are helping us from washing away this gradient. All else being equal, we have blood flow moving through our capillaries, right? Exchange happens in capillaries, so it's possible in a different anatomical arrangement that they just kind of move excess water and salts out and take us back to kind of homeostasis, take us back to an evenly distributed kidney. But because of the shape here, where we have a descending limb of the vasa recta that's losing water and at the same time dragging in solutes. And then as blood flow goes up through the ascending limb of the vasa recta, we have water moving into the plasma, solutes moving back into the interstitial fluid, back into the kidney. Right? This helps us just not create washout here. So if you're curious, you can see the exact osmolarity of now the blood flow. So this is now looking at the vasa recta rather than the filtrate itself. So be careful there. These images look kind of similar. But you can see that osmolarity of both our blood in those vasa recta as well as that osmolarity of the medulla here listed in the center. Okay. So you can see that we have water movement out in this descending limb, but solutes are moving in. And then the opposite thing is happening in this neighboring region. Okay. So overall, what we care about, right, is that we set up this osmotic gradient. The mechanism by which we set it up with those two parts of the loop of Henle, that descending limb and the ascending limb, that's called our countercurrent multiplier. Right? Now we're going to be using this osmotic gradient to do regulated reabsorption. Okay. So as we look at regulated reabsorption, we're going to see that the distal tubule and collecting duct are sometimes going to be permeable to water and sometimes not. So sometimes osmosis can happen and sometimes it can't. And that's dependent on the proteins embedded in these cells and the degree to which they are or are not present. So we can think of them as water channels, and they're called aquaporin 2 and aquaporin 3. So when we think about these structures, I'm going to draw us a little cell. All right, so we're going to have a flat basolateral membrane, which would be on the blood side. And we'll get a squiggly apical membrane towards our filtrate. Right? We're saying that when we're looking at these aquaporins, right, in the basolateral membrane, you always have an aquaporin 3 for water to go through. This is our aquaporin 3. Okay. So water is going to be able to move across this membrane, right? So water in the cell, for example, could go towards the bloodstream, right? But it's critically the presence of aquaporin 2 that's going to control whether we can actually reabsorb more water here, right? Because you'll notice, if you don't have a water channel here on the apical membrane, right? Aquaporin 2 is the one that would be there. It doesn't really matter if you have aquaporin 3 on that basolateral membrane at all, right? Without an aquaporin 2, the water just kind of turns around and stays there, right? So what we're going to be paying attention to as we look at regulated reabsorption is that presence of aquaporin 2, right? Because when it is present, we're going to be able to have water move all the way across the cell, right? So we'd first see water crossing into the cell through aquaporin 2, and then exiting towards the blood, towards the plasma through aquaporin 3. So to actually have water reabsorption, we're going to need to have both of these present to go all the way across the cell. This presence or absence of the aquaporin, specifically aquaporin 2, is going to be controlled by our hormone ADH, or antidiuretic hormone. Okay. So we saw the real picture of our distal tubule and collecting ducts here, right? So a little kind of scragglier looking apical membrane that I just drew it, but we know that we have these tight junctions here sealing across. This is important because it means water can't like sneak through between cells. Okay. So now we're looking at what happens when these aquaporins are present or absent. Okay. So we're going to see that when our late distal tubule, which you can see up here, right, and our collecting duct, which is what's going down here, right? When they are impermeable to water, right? So no aquaporin 2, so we can't have any movement of water into these cells. This means that water can't leave the tubules at all, right? So that means the osmolarity of our tubular fluid, and then therefore our urine, it's just not going to change as it moves through these regions. There's nothing left that can be moved when we have this kind of sealing over of these water channels, right? So our tubular fluid, as it goes through the late distal tubule, it is at 100 milliosmoles for its osmolarity. And if you want to remember how it got there, we had a sort of series of images that you can look through, seeing how that osmolarity of tubular fluid changes. But otherwise, just know it's at 100 here, right? So we know that there's a potential pull on water from the renal cortex here. It's at 300, so that's a higher osmolarity. But without these water channels, without these aquaporins, can't move water, so we just see that same osmolarity continuing through the collecting duct. We see just 100, 100, 100 over and over and over again until we eventually make our way to the bladder as urine, right? Because water can't move. So when water doesn't move, when we don't have more water being reabsorbed here, this means you're going to end up with urine that's pretty dilute and pretty high volume, right? You've got lots of water left in this filtrate as it goes on and becomes urine. Yeah. Now, when we have our hormone ADH present, we stimulate the insertion of water channels onto that apical membrane. So we put the aquaporin 2s into the apical membrane, and now we're going to let water flow all the way through the cell. So across that apical membrane, and then again across that basolateral membrane through the other aquaporin 3. So we are able to have osmosis happening fully all the way across the cell. So we are able to reabsorb water. So as we descend through the medulla, we're going to see that urine getting concentrated and concentrated and concentrated until we hit our maximum urine concentration, which is 1400 milliosmoles, right? That's the same number we got to before when we were looking at the countercurrent multiplier, because this maximum amount of water reabsorption is dependent on that loop of Henle and how deep it goes into the kidney, right? We're setting up this maximum urine concentration. And the most we can do is become isoosmotic, so come to equilibrium with those deepest parts of the kidney. Okay. So on this next page, right, we see the situation on the left where we have a distal tubule and collecting duct impermeable to water. So this is our no ADH version. Okay. So we can see that we have that low osmolarity, high volume of urine as we leave the kidney, right? So we're at 100. But on the right-hand side, we see what happens when we do have ADH present, right? So yes to ADH. Okay. So now we see that as we go through the distal tubule, right, now we take advantage of this gradient between our tubular fluid and the cortex, right? So we have water moving from that osmolarity of 100 towards this region where we have an osmolarity of 300, right? Which means we see the urine starting to concentrate, right? So we can see that now we're at 300 milliosmoles as we start to go through the collecting duct and dive from the cortex to the medulla. And you can see this number gets higher and higher and higher as you go deeper, because as you go deeper, you have more and more and more water leave towards these more highly concentrated regions of the kidney. Okay. So more and more and more leaves until we come to equilibrium with that maximum osmolarity, right? And so at this point, we've moved as much water out as we can. So we're going to have this high osmolarity of urine as well as a low volume, because most of the volume of your urine was the water, right? And that's actually why we call this hormone anti-diuretic hormone. A diuretic maybe you've heard of like coffee being a diuretic. A diuretic is something that makes you pee a lot, right? So this is anti-diuretic. You don't pee so much, you have this low volume urine because you're reabsorbing all the water. So the water isn't going to your bladder, it's getting funneled back into your bloodstream. Right. So this is how we also get to that obligatory water loss that we mentioned briefly before. So the maximum osmolarity our urine can have is controlled by this medullary osmotic gradient. So maximum is at 1400. And we do still have a little bit of water in that urine at 1400 milliosmoles, which is good because we need about 440 milliliters of water to leave your body as urine in order to be able to eliminate all of the solutes, all the waste products that you are producing day to day. Okay. So we're going to take a deeper look now at ADH and how it really works. Okay. So our goal by sending out this ADH signal to the kidneys is going to be to increase water reabsorption, which means we're taking water out of the urine essentially and pulling it into the blood. Okay. This is happening by controlling how permeable those late distal tubules and collecting ducts are. So controlling whether water can move or not. Okay. And therefore controlling the osmolarity of the urine. Right. So 100 if we have completely impermeable distal tubules and collecting ducts. And at our maximum amounts of ADH, we're concentrating all the way up to 1400. Right. And the way we're doing this is by varying that presence and absence of aquaporin 2. Okay, so we're going to walk through this chart. What we're looking at is a principal cell, that's just a type of cell, lining our late distal tubule or our collecting duct. So what we're going to be looking at is what happens when we have ADH coming down our bloodstream, and how does it act on these cells? Okay. So we're starting out by noticing that we have in this image our hormone, ADH, right? So antidiuretic hormone is going to be present in the plasma, starting all the way down actually from your posterior pituitary. Okay. And once it's in your bloodstream, it can pass through our capillary endothelial layer and through this peritubular fluid and come into contact with the basolateral membrane, so that base of the cell. So we can see here that we have a receptor for ADH. So it's a G protein coupled receptor. So we know that those tend to have kind of complicated multi-step processes when we see G protein coupled receptors. And this is going to hold true for this ADH receptor. But what's happening first? ADH is coming into contact with that receptor. Okay. So when it does, we have a reaction. So our G protein splits apart. That's just how G proteins work when they're attached to our receptors. And we can see here we're activating this cyclic A&P pathway within the cell. So this is starting out our second messenger system of signaling inside the cell. Okay. There is, of course, a pathway when we're thinking about our second messenger system here. So our cyclic A&P then goes on, activates this protein kinase A. Okay? But what we really care about here is that what this is going to do is this is going to trigger these aquaporin 2 molecules, these aquaporin 2 channels we'd actually already made. We have them sitting around kind of waiting inside the cell and vesicles. So we have those there. And what this is going to do next when we get this signal from this second messenger system is that we're going to take that vesicle and we're going to pop it on the apical membrane so that those aquaporin 2s are now present, right? So this is how we inserted them onto our apical membrane. Okay. So we can go through those steps, right? ADH heads down our bloodstream, activates this receptor. We then have a cellular pathway that tells us to take these aquaporin 2s put them on the apical membrane. And that means that we now have a pathway for water to flow first into our cell through aquaporin 2. And we already had an aquaporin 3 on that basolateral membrane. So the water can continue to flow back towards the plasma. Okay. So this is how we are controlling our excretion rate of water tied to that reabsorption rate for the water. And so we want to be able to tell, what does our urine look like? What is our reabsorption? What is our excretion when we have this hormone present versus when we don't? Okay, so we're going to start out, right? When we have ADH there, when we've got ADH present, are we going to see higher or lower water reabsorption? And are we going to see higher or lower water excretion under these conditions? Okay, take a look. Okay, great. Yeah, so when ADH is present, we're trying to do more water reabsorption. Perfect. We insert those aquaporin 2s, higher water reabsorption, which means that we have less water in the urine left to excrete out. So we've got this trade-off between water reabsorption and water excretion. Perfect. Okay. So when we're thinking about ADH, we might also want to know a couple of details about where it's coming from, because as we walk through a little further, we're going to see that there are some interactions between ADH secretion and secretion of some of our other hormones. Okay. So it's important to know that your body is basically always sampling a little bit of your blood, sampling its osmolarity. Okay. So we have these osmoreceptors in your OVLT region. So you can think of them as kind of like taste testing your blood as you're looking at the brainstem, sensing its osmolarity. So sensing basically how concentrated it is, how many solutes we have relative to the amount of water. Okay. And in response, when these receptors sense that we don't have enough water in our bloodstream, so if it's getting kind of too salty, we're going to be releasing is ADH from our posterior pituitary, right? So remember, we talked a lot about the pituitary when we talked about the endocrine system. Almost everything we talked about was from the anterior pituitary. So here is pretty much the first time we are focusing in on a posterior pituitary hormone. That's where it's coming from. Okay. So this OVLT is special and especially able to sense these properties of the plasma because unlike other parts of the brain, it's not surrounded by our blood-brain barrier. So it's able to come more closely in contact with the contents of our blood. So that's part of why it's able to trigger this reaction from the posterior pituitary. The other way that we can release ADH or know that we need to release ADH is by sensing our blood volume and blood pressure, right? Because fundamentally, when we're reabsorbing water, right, which we would do if we have ADH present, what we're doing is we're increasing blood volume, maintaining blood pressure by putting enough volume there, right? So this goal here is to reabsorb water into the bloodstream. So when we see changes to blood volume and blood pressure, especially when we see low baroreceptor activity, this is going to increase our secretion of ADH in order to kind of resolve the situation. Okay. So here we're looking at some of those pathways in a little flow chart. Right? So what we're looking at is a situation where we have high osmolarity in our extracellular fluid. So plasma is one of our types of extracellular fluid. So this can be our bloodstream, right? And we have this reaction in our brain that's detect and respond, right? So we're going to see that we have this detection of high osmolarity, and then we see our hypothalamic cells responding, so increasing their activity. And the goal here is to increase our ADH secretion, which means we increase our water reabsorption. So we're going to be keeping water in the bloodstream, keeping water in that extracellular fluid as we decrease our water excretion. Don't pee it out, conserve body water to try and keep that osmolarity down, which in essence is going to keep blood volume up as well. So here we see essentially the same pathway, just showing how that plays in with MAP and with blood volume. All right, so we can see we're still talking about a situation that causes ADH secretion to increase water reabsorption and stop our water excretion, right? But in this case, we're looking at our drop in blood pressure and our drop in blood volume, right? So in these situations, we have low blood pressure, low blood volume. We have our baroreceptors that can sense this. So similarly, they activate these hypothalamic neurons that then communicate with the posterior pituitary, telling it to release ADH. Okay. So when we're thinking about that urine, we can think about it with relationship to these kind of higher up levels too, right? So if we have urine and we're looking at its volume and osmolarity, when we've activated these receptors, when our posterior pituitary, when we haven't activated these receptors and our posterior pituitary isn't secreting ADH, what's our situation? Sorry, guys, I'm reading it backwards to you. Let's take a look. Okay, good. Yeah, so if we don't have ADH being secreted from the posterior pituitary, that's our situation with high volume urine because we don't have these aquaporin 2s without ADH. And this means when we have high volume urine, it is going to have low osmolarity. I can point out to you that you're always going to see a trade-off in urine between volume and osmolarity. They're always going to be opposite of each other, right? Because if you have a lot of urine, right, it means a lot of water, not much solute to balance it out. You're not really going to see high volume urine that also has high osmolarity. It's always going to be when we have that really concentrated, low volume urine that we see that osmolarity start to build up. Okay. So when we think about regulating water excretion, primarily we're going to be thinking about ADH and controlling, regulating that water reabsorption by changing the presence and absence of aquaporin 2. But we do also see changes to water excretion as we see changes to glomerular filtration rate, right? Hopefully, our glomerular filtration rate is being appropriately regulated. Reminder that we have this central range between 80 and 180 millimeters of mercury for our mean arterial pressure, where we shouldn't see a change in GFR. Right? But if we see drops below this range or increases above this range, your kidney kind of can't self-regulate outside of this regulated range. So we do see changes in our filtration rate. Right? So if you see a super low drop in blood pressure, right, a big force for filtration, for movement of fluid into our nephron, into the kidney, was that hydrostatic pressure in our glomerular capillaries. So if we have low blood pressure in general, we also have low blood pressure in that capillary. which means that we see filtration slowing down. So low blood pressure means we have a decrease in our glomerular filtration rate, which means there's not as much water in the nephrons, which means by the time we leave the nephrons, head to the bladder, we're going to see less water, right? So less urine, less water excretion. But when we end up with really high blood pressure, you may have noticed, perhaps if you know anyone with high blood pressure, these blood pressure spikes can cause increased urination, increased water excretion, right? You might even have noticed this if your blood pressure is spiking because like you're really nervous, right? Your sympathetic nervous system really active, you're stressed, you're fight or flight. You might notice you have to pee all the time, right? This could also potentially have to do with blood pressure, although hopefully you're staying under 180 for your mean arterial pressure. But we can think of it conceptually that way. Right, so if we have a really high blood pressure, it's going to increase that glomerular filtration rate. So lots of water is going to be pouring into the nephron. It's going to be whooshing fast through it. And so we'll see an increase in water excretion ultimately. So we shouldn't see these because we should be regulating that mean arterial pressure, but we might. Our final topic is our sodium and potassium balance. Okay, so this is going to work kind of in sync with our water regulation. And these ideas of balance are the same when we're thinking about solutes as when we were thinking about water. Okay, so we want to maintain plasma concentrations of sodium and potassium as well as the volume of water. And so we have words for when these are out of balance. So what you're going to notice when we're looking at these sodium words, I really like that they put Na in the middle for us, super helpful, right? Because that's our ion, right? Sodium is Na plus, okay? So when someone has hypernatremia, what we're saying is that their plasma has lots and lots of sodium in it. Hyponatremia means we've got low plasma sodium. Okay? Sodium we know is super important because we talk about it all the time. It's the main solute that we see in extracellular fluid. We know that we have sodium potassium pumps all over the place. We know that we need a sodium gradient for excitable cells like neurons and muscles, right? Action potentials. This is also going to be important for just normal osmotic pressure, normal flow of water across cells, okay? So keeping sodium balance can also be important, okay? So if we're thinking about tracking sodium through our kidney, as we're moving to the kidney, we're going to see that at the glomerulus, at the renal corpuscle, that sodium gets freely filtered, right? So sodium, like water, like everything that's not a protein, just goes through into our nephron, the same concentrations essentially as in our plasma, right? So not blocked by anything. Okay? And so then it's there to travel through the proximal tubule. So in the proximal tubule, we see a lot of unregulated reabsorption like we do for other solutes, like we do for water. Okay? And we're going to see more reabsorption of sodium as we get to the distal tubule and the collecting duct. Sodium isn't a substance that we're going to think about being secreted, so we don't have to worry about that extra little detail when we're balancing out. We're really just focusing on how much came through initially, how much was filtered through initially, and then how much gets reabsorbed. And that reabsorption, when we get to the distal tubule and the collecting duct, so the latter parts of the nephron, is going to be regulated now, right? So reabsorption does get regulated, right? And it's going to be regulated by these two hormones, aldosterone and ANP, or atrial natriuretic peptide. Okay. So this is going to happen in the same sorts of places where we see ADH regulating water reabsorption. So distal tubules and collecting ducts, specifically in a type of cell called a principal cell. This is going to be active reabsorption. It is going to use energy because we are going to be using a sodium potassium pump even though we're also going to see the need for these signals from elsewhere in the body to tell us it's time to do this. Okay. So here we're looking just at the proximal tubule first, in case you're curious about that sodium reabsorption that's unregulated, right? So no hormones anywhere in this image, but we do see that we have sodium potassium pump, which would use ATP on this basolateral membrane. And we know that sodium is traveling into our proximal tubule epithelial cells in a bunch of different ways, right? So sometimes it's helping us carry other solutes with it. Sometimes it's doing counter transport with hydrogen ions. But overall, just we know that sodium is following this gradient set up by the sodium potassium pump to ultimately be reabsorbed into the plasma. Now, when we get to the distal tubule epithelial cells, we're going to see some differences and some similarities, right? So conceptually, we're going to see some stuff that's quite similar, right? So we are still going to see sodium potassium pumps, right? We might see some co-transport, right? So moving along with something. We're also going to see that we have sodium channels that are just individual sodium channels moving on its own across an apical membrane. Okay. So we're going to think about aldosterone first. Right? We got aldosterone and we got A&P when we're thinking about effects on sodium. Okay. So when aldosterone is present, this is going to increase how much sodium reabsorbs. So we're going to move sodium into the plasma in the presence of aldosterone. So this is another hormone, but this one isn't coming from the pituitary. This one is actually coming from your adrenal cortex. So remember those adrenal glands sitting on top of your kidneys, right? Aldosterone is coming right there from those glands that are pretty close to us when we're looking at our kidney. It's acting on principal cells in the distal tubule and collecting ducts. And the way it's working is really just, it's going to increase how much sodium and potassium gets pumped across the basolateral membrane. Right, so we're working faster, basically here, creating a really strong sodium gradient and potassium gradient using ATP there. And then to use up this strong gradient that we're creating, it's going to increase the number of open channels that we see on the apical membrane. This is going to have an effect on sodium. It's also actually going to have an effect on potassium. But when we look at potassium, we're just going to want to make sure that we are actually tracking potassium's gradient, where potassium is high and low, because it's always pretty much the opposite of sodium. So if we're looking at the sodium, right, we see that we made a gradient where we have really high sodium here towards our peritubular fluid, really low sodium inside this principal cell. And now we've opened up these sodium channels. So we see sodium moving from a higher concentration in our tubular fluid, entering towards that low sodium region, and then getting pumped out towards the blood. Okay, so potassium just does the opposite, moving out of the cell, right? Getting pulled from this basolateral membrane side and then distributed into our tubular fluid. Okay. All right. Now, when we think about aldosterone, aldosterone is actually part of a much larger system, okay? So we have a flow of messengers, essentially, that control the amount of aldosterone that gets released by our adrenal cortex. So when we're looking for what's going on with aldosterone, we're going to see that this is linked to two other hormones, essentially. So we're going to have renin, we're going to have angiotensin, And then we're going to have aldosterone. And this is referred to as the RAAS, so our RAS system. Okay. So these are all coming from different places, some of which are in the kidney, some of which are not. Okay. So we're going to think about this renin first. Okay. So when we're looking at this system, we're going to see that renin comes from our juxtaglomerular cells in the kidney. So we saw those juxtaglomerular cells before as part of our juxtaglomerular complex. So we remember they were on this image when we were looking at what's passing by next to the glomerulus, basically, but communicating with the kind of end flow of our tubular fluid. Okay. So these juxtaglomerular cells can secrete renin. Okay. And when we have renin in the bloodstream, we're going to see that renin and angiotensinogen work together. Okay. So we can see here that we have endothelial cells with an enzyme on them. So ACE here is our angiotensin converting enzyme. Okay. So what's happening here, right, is that we see our liver secreting angiotensinogen. Right. So this would be kind of our first step. Right. Right. So first step, we're secreting angiotensinogen from the liver. Second step, those juxtaglomerular cells have to be releasing the renin. Okay. And then our third step is going to be the conversion of angiotensinogen to angiotensin 1. Sorry, the 2 is not in between, but here we have 3. So renin is the thing that's doing that. All right, so renin was step 2. Okay. And this angiotensin 1 bumps into the sides of the capillary, contacts that ACE enzyme, okay, and gets converted into angiotensin 2. So angiotensin 2, you're now seeing the plasma here. Okay. And it's actually that angiotensin 2 that ultimately stimulates the creation of aldosterone from our adrenal glands. All right, so that's kind of step 5. Okay. So in order to release aldosterone, we have to communicate between a bunch of different systems. The liver is kind of always producing inactive forms of proteins. So we notice that inogen ending on angiotensinogen kind of gives us an inkling that this might be something that has to be activated, like when we were looking at our clotting factors coming from the liver. Right, so angiotensinogen is kind of always swimming around, but we need this renin to convert it into angiotensin 1. We got capillary endothelial cells with their enzymes, need to convert again to angiotensin 2 before we can trigger aldosterone. Okay. Oh, so here we have that zoomed in for you, right? So we made our angiotensinogen with the liver, right? Needs to come into contact with renin from our juxtaglomerular cells, right? Renin is going to be responsible for creating that angiotensin 1. Okay. Our capillary endothelial cells then help us convert to angiotensin 2. And that's when we can finally make our aldosterone. Okay. So a bit more complicated, I think, than how we made our ADH. Okay. So when we think about how angiotensin 2 works, right? So we're zooming in here, angiotensin 2, right before we hit the adrenal cortex and make the aldosterone. We'll see that we actually have kind of a bunch of things that it does, right? So what we've just added on to our idea of angiotensin 2 is this interaction with our adrenal cortex and the creation of aldosterone. But you might remember, or you might not, that we've had angiotensin 2 actually kind of on some of our extra little detailed bullet points through this course as a hormone that's vaguely involved in things, and we weren't going to focus on it at that point. It is on this chart here, right? So angiotensin 2, another effect that it has is vasoconstriction, right? So working kind of along with our sympathetic nervous system that way, right? And we're going to see that it also plays a role in helping us secrete ADH, and it even plays a role in making you feel thirsty. Okay, so that's because our big role overall here, right, our big goal of all of these processes is ultimately to raise blood pressure, right? So usually when we're creating this angiotensin 2, because we're creating renin from those juxtaglomerular cells, converting it to angiotensin 1, and then ultimately to angiotensin 2, like we're doing that because we're noticing some kind of low blood pressure situation, right? So one way we can fix low blood pressure is just to squeeze down arterioles. So angiotensin 2 does do that, right? So increasing resistance to increase mean arterial pressure, right? I'm going to jump to the bottom corner here before we do this complex bit in the middle. But another way that we can increase blood pressure, right, is to increase plasma volume, right? So increase the amount of fluid, right? So we can do that all the way to the right-hand side here is just by drinking more, right? This is why angiotensin 2 has this interaction with thirst, right? Makes you thirsty to try and get your plasma volume up, to get your blood pressure up. And these two blocks in the middle here can work on their own, but really often work together, right? So we understand, hopefully, why ADH and this increase in reabsorption increases plasma volume, but might not immediately be obvious why sodium does that too. So to do that, we'll focus in right here, right? So what we see happening when we see the secretion of aldosterone from those adrenal glands, when we reabsorb the sodium into our bloodstream, right, into the plasma, we're not like done yet, right? So you took a bunch of salt into your blood because we had this aldosterone around, right? We inserted sodium channels into those apical membrane. But now your blood is salty, right? Now we have high osmolarity of our extracellular fluid, like our plasma, right? So that was the trigger for ADH secretion. So usually when we see aldosterone secretion, we're going to see this kind of 1-2. Aldosterone will drive sodium reabsorption, but then all that sodium is going to tell your body, hey, I need to absorb a bunch more water too. So these kind of all go together. Okay, so I see we're at time. So we will